Hello, hello, hello. Module 5, Recitation 6, Central Dogma. Very important concept when it comes to general genetics or genetics in general, right? So these are some pretty important concepts to understand. So let's go through these questions one by one and figure out how to solve them. So for this first question, it says, which of the following are not DNA, are not a valid DNA sequence? So we want to remember that there is a few main differences between DNA and RNA. So if we check out these slides from your recitation, which you guys have available to you, we've got the history. We'll talk a little bit more about that later, but let's just look at some of the differences in DNA and RNA. So with DNA, we see our 5 carbon ring right here, our sugar ring. And then we see that we have these different nucleotides. And so for the most part, DNA and RNA are pretty much the same, but some main differences here are that DNA has a deoxyribose, which is, so we're seeing, so these are hydroxyl groups. So for anybody who has not taken organic chemistry or um, OH is a hydroxyl group, so the structure is completely the same across the board. Here's a hydroxyl, except for on this carbon here, we have a just a hydrogen versus a hydroxyl group. So that's why it's called a deoxy. So less one oxy, oxygen, ribose, and ribose because this is a sugar. This is a pentose sugar. And then, so the other big difference is that DNA uses thymine for this particular nucleic acid, whereas an RNA has a uracil, U. So T versus U. So if we're looking at these structures here, there are some slight differences in how thymine and uracil look, but the important thing is that they are both able to make these two hydrogen bonds between adenine and the uracil or thymine. So, main differences: deoxyribose, it has one less oxygen, and the nucleic acid will be thymine with DNA or uracil with RNA. So let's go back to our quiz. So which of the following sequences is not a valid DNA sequence? So really we just need to think about, where are we seeing the differences in thymines and uracils? So we know for a fact that DNA will not have any uracils. So if we look at these, the first answer says these are all possible DNA sequences. If you check out each of these sequences, I think you'll be able to see that there is one that could not possibly be a DNA sequence based on the information that was just given. What makes DNA replication semi-conservative? So let's remember what the difference is between semi-conservative, conservative, and dispersive models are. So here we have this, the models of DNA replication. So if we're looking at a conservative model, what this is saying is that both of these strands will be replicated, but when they come together to create the two daughter strands, one DNA, double helix DNA, will have both the parent strands, hence the dark blue, and the other double helix will have both the daughter strands, which is the light blue. So that's the conservative model. The semi-conservative model, which is what we know to be the most true, that's what we believe to be the way in which things work. And if you'd like to review why we know that, or you can go ahead and double check some of the past experiments that have proven this to be true, specifically with a carbon. So if we're seeing here, we have these parent DNAs, then both strands are being replicated. And in this one, we see that there is a dark blue and a light blue. Remember, the light blue is the daughter strand. So in this semi-conservative model, what we're seeing is that there will be one parent strand and one daughter strand per daughter DNA. And then the reason why you're seeing these ones that have just two daughter strands is that if you pull these two apart and replicate it, they will both be daughter strands if you want to consider it that. And so this parent strand can only go to one of these two, and the other one will have two of those daughter strands in that second generation. And then dispersive model, what dispersive model says is that we replicate in pieces. So there'll be a replication here, a replication here, a replication here, a replication here, and it'll be sort of cut up. And so what we see are these chunks of parent and chunks of daughter, chunks of parent, chunks of daughter cells, strands, excuse me. So the thing to remember about this is what this is saying is that we're only replicating small pieces at a time. We're having to literally break apart the DNA, replicate that section of the DNA, and then puzzle piece the DNA back together. And so, hopefully, with that description of how this dispersive model would actually have to work, you could see where there could be a lot of issues along the way. So, that's our review of the different models of DNA replication. So when we're looking at what makes DNA replication semi-conservative, so again, semi-conservative is this one parent strand and one daughter strand per second generation, so to speak. So let's look over these answers and see which ones you think follow that. So one strand is longer than the other. There's no really reason to believe that. Material is distributed randomly between both strands. I feel like we could probably say that that's a dispersive type model, right? Both strands have a portion from an original copy of the DNA. And then both strands are completely new material. So I think if you look at this again, we can see that what we would consider a semi-conservative model. So, question 3, which of the following would be a stop sequence? And so remember, let me just pull this up real quick, and I'm sure you guys have access to this. And so these are the amino acids. This is just an amino acid chart. You can look it up anywhere on Google, super easy. But there are 3 stop sequences, and there are some acronyms to help you remember it. They all do start with the letter U. And they do only contain the letters U, A, and G. There is no C's in the stop codon. So, so yeah, it's not a bad thing to try to remember UAA, UAG, and then UGA. Um, so if we were looking at that, you know, if you pull that up, you'd be able to figure out which one of these is a stop sequence. So based off Chargaff's rules of base pairing, if there is 21% thymine in a DNA strand, what percentage would there be of guanine? So for Chargaff's rule, what Chargaff discovered was that, and here's all your Messelson and Messelson and Stahl experiment. I'm sorry, I said carbon. I meant to say nitrogen. This was how we discovered that it is a semi-conservative replication. And so just review this to understand why. But if we wanted to look at Chargaff's rule, what Chargaff's rule says is that there is an equal number of bases of complementary base pairs to each other. So there will be equal number, sorry, I don't see Chargaff's rule in this. So there will be an equal number of, okay, here we go. There'll be an equal number of adenine to thymine or adenine to uracil. If we're talking about an RNA sequence, you're going to have an equal through replication transcription, you're going to have an equal number of uracil per adenine that you have. But as far as in a DNA sequence itself, you are going to have the same amount of adenine as you will thymine, and you will have the same amount of guanine as you will cytosine. And so it won't necessarily be 50-50, like you'll have 50% adenine and thymine and 50% guanine and cytosine. It just means that the percentage of these two will be equal and the percentage of these two will be equal. Um, so for this, you can look at if there's 21% thymine in a DNA strand, what percentage would there be of guanine? Well, we know that guanine and thymine do not base pair together. So an easy way to think about this would be to, let me just get my annotation out. So we could, if we have A to T, and we have G to C. We have 21% here, which means we will also have 21% here. So if we add those two together, that equals 42% total. And obviously, 100% of the DNA is made up of all four of these. So if we do 100% minus 42%, whatever this equals is going to be the combination of these two. And because these two are equal to each other, then we know that this percentage here is going to be split equally between each of these, G and C. Okay, so that would be the best way to figure that out. And central dogma states that DNA turns into RNA and RNA turns into protein. This is not what the central dogma states. So it's really important to understand that the central dogma uh, DNA is translated into RNA, mRNA technically, but and then mRNA leaves the nucleus into the cytoplasm and is transcribed into a protein. But this chunk of DNA that is used to write an RNA sequence does not itself transform into RNA. So, what we mean by that is in this sequence, we don't take this chunk, switch this out for uracil, and then transfer this whole thing out of the nucleus. We break this in half and we use a ribosome to transcribe each side, but let's just talk about this one side. So transcribe this side of the DNA from 5' to 3', and then we take that out of the cell, and then that message is, that's why it's mRNA, messenger RNA, is used by ribosomes to translate it into protein. And we use tRNA, which is a transport RNA that brings these amino acids to the ribose that's doing the transcription, the translation. Okay. So not turning into. mRNA is read in the cytoplasm by, so let's look at, oops, didn't mean to do that. Let's look at DNA to mRNA. Let's see if we can find an image to show us what this looks like. Do, do, do. So what we are seeing is, well, these aren't very good images, I won't lie to you. But here's a nice image of how it all kind of goes along, right? So we have this DNA, we have our RNA polymerase that comes along and creates our mRNA. And then remember that we have it's pre-mRNA because it's before we do any post-transcriptional modifications. So we have our poly A tail, our M capping. We splice out things in eukaryotic cells. We're able to use the same section of DNA to create a bunch of different proteins. So we have introns and extrons and all that good stuff. So anyway, that's what actually leaves the cell. And then this piece of what's called mature mRNA comes out of the into the cytoplasm, out of the nucleus, and is translated by tRNA and another set of ribosomes. Every like the ribosomes always work in pairs, so they have one that kind of grips on to the sequence and another one that couples on and moves things through. And so we have our APE sites, and that just talks about what is happening along the way. So an amino acid, this is a tRNA here that has an amino acid attached to it. It comes into the ribosome at the A site. There's some cool science stuff that happens that connects making this polypeptide chain, right? So from one peptide to another, and as we increase, it's called poly from multiple polypeptide chains. As it's coming out, there'll be some modifications, some twisting, bending, connecting, shaping that happens, protein folding, right? And then as the tRNA leaves, it goes to get another amino acid, and this chain gets longer and longer and longer. Here we have this polypeptide chain. The protein folding usually happens as it comes out. It doesn't always happen later. And then we have this functional protein. So this would be called your post-translative post-translational modification. So this is a good thing to look at here as far as like why and how that works. As far as um, let's see, translation, oops, transcription. What we are seeing is, I wish. I was trying to find a good picture for you guys. Um, but essentially, if we wanted to look at the differences between these, so topoisomerase actually keeps it from supercoiling. The as we unwind the DNA strand will have this supercoiling effect outside of that break. And so we see, let me see, maybe if I look at topo here, this is a good picture for this. And so we see that, here we go. So topoisomerase keeps it from uncoiling, and or keeps it from that supercoiling outside of this breaking. And then so we have here, this would be, we would also have um, our helicase is what is going to split these open. So it will move. It's like this little triangular thing that moves it this way, up and out of the way. And so what we're seeing here is we're seeing like a polymerase chain, that a polymerase that's going to be doing the translation. So that's what we're seeing here. Um, do, do, do. So yeah, and ligase is what helps keep this together. Um, so ligase is what will glue some of these things together. So it'll bind things like it's used in like replication and repair, doing like the Okazaki fragments, keep getting these fragments together. So that's what ligase will do. Um, So for our question 7, one codon will code for several amino acids. We know that to not be true because we see here one codon, UUU, only codes for this amino acid here. This codon is not found anywhere else on this map, right? Anywhere else there. So we know that that can't be true. And actually, it's the opposite of true because what this is saying is, sorry, what this is saying is that we actually have multiple codons that can code for the same amino acid. So we're saying that this is actually the opposite of true. And then for question 8, given the image below, after 10 generations, you centrifuge a sample of genetic material, which tube would reflect a dispersive model of replication. So blue is 14, red is 15, and a burgundy purple is both. Okay, so let's just look again real quick at what this dispersive model looks like compared to all of these other ones. If we were to look at the DNA, it looks like a mixture of both parent and daughter cells. Each DNA itself is a mixture of parent and daughter. So as we go further down, we're seeing this mixture model all the way down. Whereas in this one, we're not seeing that. We're seeing this one parent strand that goes all the way down and all the rest are daughter. In this one, we see parent strands and we see daughter strands, but per DNA strand, this one is a very mixed model. So if we're looking at this and we wanted to consider which one looks like the most mixed model, well, if purple and burgundy represent a mixed model, and we know that with a dispersive model, we are going to have a mixture of DNA per strand. I think it would be best to say that the most purple would be the most representative. So for the next 8 questions, we'll assess whether the statement is in regards to DNA, RNA, or both. So the structure replicates semi-conservatively. We know for a fact DNA does. We've already talked about that. Does RNA replicate semi-conservatively? RNA does not replicate. RNA, if we're talking in RNA can be used to create DNA, but we do not. RNA is actually it it breaks down pretty quickly. It's not a very stable molecule or a structure, and so we don't. It's never used to create more RNA. How we make RNA is with DNA. So with question 10, the structure contains 2 hydroxyl groups. So let's just real quick look at this one again. So again, a hydroxyl group is OH. And so we see that the structure has 2 hydroxyl groups. I'm sorry, a 2' hydroxyl group. My apologies, not 2 hydroxyl groups. A 2' hydroxyl group. So this is 2'. So how you would count on the carbon would be 1' is what position carbon it is. So 1', 2', 3', 4', 5'. Those are that's how this is a pentose sugar, a ribose. So on the 2' carbon, there is a hydroxyl group. The structure contains guanine. So in here, we know that DNA for sure has guanine. As far as RNA, the only difference between DNA and RNA is the thymine to uracil. For number 12, the structure contains uracil. DNA does not contain uracil. Only RNA contains uracil. The structure carries information out of the nucleus. We know that RNA comes out of the nucleus in order to be translated into a protein. DNA, on the other hand, is a well-protected genetic book, story, sequence that does not come out of the nucleus. It is in the nucleus to remain protected. There are a lot of safeguards in order to enter or exit the nucleus because it is such a critical part of survival of any living thing. The structure contains thymine. We have sort of hit ourselves on the head about what the difference between DNA and RNA is when it comes to nitrogenous bases. The structure contains a deoxyribose, DNA or RNA or both. So deoxy means less one oxygen. So less one oxygen. And last but not least, the structure contains 5' and 3' ends. So during replication, we see there is 5' and 3' end um, here on our DNA structure. And really what all this is saying is 3' and 5' end is all we're doing is identifying where the phosphate is. So on our sugar phosphate backbone, we have this connection here. And so this is our 5' end because this is our fifth carbon, right? And this is our 3' end because this is our third carbon. One, two, three, right? One, two, three. Oops, I'm sorry, I counted the wrong way. One, two, three, four, five. Right? So this fifth carbon is out here on this arm. And so that's why this is a 5' end. Now, again, remember that there is no difference in structure with DNA and RNA. So there will be still a 5' and 3' carbon on both DNA and RNA. If you have any questions, please feel free to reach out. I have also included some really interesting articles about the discovery of the DNA structure and how, we came to understand DNA. I just would love to, um, put out a big shout out to Rosalind Franklin, who is actually the one who first took a picture of the DNA structure. This was a very long worked at project to figure it out. And so with her X-ray diffraction, chromatography, she actually took a picture of this, and you see this X, and this is called Photo 51. And this was actually one of the first models. First photos of real life DNA that was used in order to construct that, and so there's a lot of different history on that, and so a lot of people think Watson and Crick they were the discoverers of that, but they've actually stated in written in written works that if it wasn't for her photograph, they probably would not have come to the conclusion that they came to. And so it's just really important to understand, history and where the stuff came from. So yeah, I included all of these articles here, and then I just wanted to send out one last final shout out because Dr. Watson did end up passing away just recently. He died at age 97. And so, again, another tribute to our science understanding and genetics understanding. And so it's really it's always a tragedy, of course, when these kind of founding fathers, so to speak, pass away. But so some really interesting articles here that I did include in your recordings and on your links on this page. So let me know if you need any help and have a great rest of your day.